Assalamu alaikum dear students, hope you are all doing well and you are enjoying the wonderful world of genetics. I hope previous lectures are clear to you, all of you, because this is the wonderful world of genetics. It's really awesome to teach this genetics to the students. Let's start today. We are going through another breeding experiment that Mendel performed, and that is called as dihybrid cross. That is called as dihybrid cross. Now you are well aware of the fact that the name will indicate here. Di means two. Di means two. Hybrid is when we get a result from two different varieties, and cross means reproduction. Already clear to all of you. The dihybrid cross is that type of cross in which two pair of characters are observed during the breeding experiment. No doubt there will be appearing number of phenotypes, number of characters when we are going through the reproductive experiment or the breeding experiment, but we have to consider only those selected two pair of characters which we are observing during the cross, and that type of cross is called as dihybrid cross. I repeat, dihybrid cross is that type of cross in which two pair of characters are observed during the reproductive experiment or the breeding experiment. Let's start. As I told you, first there will be parents. We will take parent plants, and again these parent plants will be pure line. Again these will be pure line. Pure line means they will possess alleles in homozygous condition, but this time we are taking two pair of traits or two pair of characters. The character which Mendel chose that time was shape of seed and color of seed. Character is shape of seed and what we call as color of seed. If you remember from the last lectures, shape of seed, dominant shape of seed is round. Dominant shape of seed is round. And dominant color of seed seed of pea plant is yellow, fine. Then recessive, that means it's contrasting plant showing the contrasting traits of same character will be wrinkled and green will be color of seed. Two characters here are shape of seed which is round and another one is wrinkled, and another character is color of seed. Color of seed is simply dominant one yellow and recessive one green, fine. Two characters are shape and color of seed, then their traits are 4. Of course traits will be 4 because we are taking two characters, fine. Representing their genotype, it will be like this: RR, YY, fine. Then here we have small r, small y, fine. You will see this is the allele pair. This is the allele pair or pair of unit factors which is controlling shape of seed. That is homozygous pair of unit factors or homozygous pair of alleles, fine. And this one, this is another pair of allele, again homozygous pair of allele, but it's controlling what we call as color of seed. Same applies here. This is the pair of allele which governs shape of seed, and in recessive form it is wrinkled shape. Then another pair of allele, Y in small case, is controlling color of seed, and in case of recessive that color is green, fine. These are the pure line parent plants. Now they are going for the cross pollination or cross fertilization, but before going for that process there will be gamete formation, because during sexual reproduction there will be formation of gametes. Gametes will be formed like this. You will see the first type of gamete will possess one allele for one character. Remember law of purity of gametes or law of segregation of gametes. each gamete should possess at least one allele for one character. One character is here shape of seed which is round, and its one allele will get into one gamete. Fine. Then another allele from the character, for the character, what is color of seed, that is yellow, will go into same gamete like this, RY. R is allele for character shape of seed, that is round, and Y is allele for the shape of color of seed, that is yellow. Then another type of gametes will be formed. They will again get the same allele combination, RY. Here we are applying law of purity of gametes. That means each gamete, when there is gamete formation, each gamete will possess only one allele for one character. These two types of gametes are possessing one allele for one character. There are presence of overall two alleles, but they are simply for two characters. One is for shape, another is for color. Fine. But again there is formation of same types of gametes. Again they are homogametes or homomorphic gametes. Same here, we get the same combination like this, small r, small y, small r, small y. In lower case, again law of segregation of gametes is followed. You are seeing that one allele is simply going into one gamete for one character. Same one allele is going into the one gamete for one character. This time character is color of seed, that is green. I hope it's clear. These are the gametes which are formed. Then after that there will be simply cross pollination. Cross pollination will be like this. First this one and this one. This plant and this plant will give its gametes, their gametes, and they will form new plant that will be like this. Fine. Then afterwards again this one, and here we will take second one. It will provide you with the same genotypic combination. After that we will take this one and this. Again we will get the same genotypic combination, and lastly we will have cross between this plant and this plant. This will be again called as F1 generation, first filial generation, first filial generation. But when we observe this first filial generation of pea plants, all the plants will produce seeds which will possess a round shape and yellow color, round shape and yellow color, because there is law of dominance followed here. That is, dominant allele will express, due to that shape of seed will be round, and here again dominant allele will express, and due to that color of seed will be yellow. All the plants of F1 generation will produce the seeds which will have round shape and yellow color. Fine. Afterwards there will be simply another step of the dihybrid cross. We will go for now what we call as self pollination. Mendel finally self pollinated these F1 generation plants. We will take this plant like this, R, small r, capital Y and small y. It will be self pollinated with itself, but we will represent same genotype here. Fine. Now before going for the process of reproduction, self pollination, self fertilization, again there will be formation of gametes. Gametes will be formed like this. Observe the gametes carefully. We will have first type of gamete, capital R, capital Y combination. Then another one will be capital R, small y combination. Then there will be small r, capital Y combination, and small r, small y. Here we are having four types of gametes. but here we were having only 2 types of gametes. Here you will see 25% gametes are this, 25 this, 25 this and 25 this. There are 4 types of gametes and they are heteromorphic gametes. They are heteromorphic gametes. They are different. They are different. This type of gamete is different from this one. This is different from this one. Every type is different. Same, this plant will provide the same genetic combination like this. Genetic combination of gametes we will have like this. These are again 4 types of gametes, again heteromorphic gametes. These are heteromorphic, these are also heteromorphic. Now we have to simply self-pollinate these plants. We have to simply go for self-pollination of these plants. For that purpose, observe the simply table which Mendel went through at the time, or which I am going to perform here. I will take these gametes here, RY, then RY, again RY and RY. These 4 types of gametes, I align them vertically. Then I will take these gametes and align them horizontally for convenience. It is just for convenience. It's like this. Fine. Now performing the cross like this. I will make a simply square. In that square I will adjust columns and rows like this. Observe the cross now. First, what you have to do, or what I am going to do, is I will take one of the gamete, this one, I will keep it constant and I will vary here gametes like this. This one and this one will be crossed. It will be like this, capital R, capital R, capital Y, capital Y. Fine. Then I will take again same gamete from vertical side, but I will vary from this will be crossed with this. It will be like this. Fine. Then again keeping this constant and moving ahead, it will be like this. Fine. Then lastly again keeping constant with, but moving ahead with the 4th type of gamete, it will be like this. Fine. This type of gamete has been crossed simply with all the 4 types of gamete. Now this is done. Move to the next type of gamete. This one keeping constant and moving ahead like this. It will give combination like this. Fine. Then again constant and this one. This one and this one. Fine. Then after this and last one. Again second is done, then taking third. This one and this one. This and third one. Lastly again keeping this constant and taking the last one, it will be like this. Again third one is done. Coming to the last 4th type of gamete, will be crossed with the other 4 types of gamete. This one and this one. Lastly this and this. This is how self-pollination was performed at that time, or will be performed even this time, can be performed even this time or in future. Fine. Now here if you observe this whole cross, we will see these types of plants will show seeds having round shape and yellow color. Fine. Again same round shape and yellow color of seeds, round and yellow seeds, round and yellow seeds. Then moving in cross, round and yellow seeds plants, round and yellow seeds plant, round and yellow seed plants. Again going upwards like this, round and yellow plants, round and yellow plants. This forms a triangle. This forms a triangle like this. It's triangle like this. These all the plants will produce seeds which will possess round and yellow seeds. Shape of seed will be round and color of seeds will be yellow. And if we simply count, it's 1, 2, 3, 4, 5, 6, 7, 8, 9. 9. We have 9 first. Fine. Then if you see this one, this type of plants, it will possess round shape and color will be green. Round and green seeds. This one. Moving here, this will show again round and green color of seeds. Then finally this one, round and green color seeds. Round and green color seeds. These will be 1, 2 and 3. We have 3 round and green seeds. 9 verbal, round, yellow. Round, yellow seeds. Then 3 we are getting round, green. Fine. Then observe this one. This will wrinkled yellow type of seeds, wrinkled yellow type of seeds, wrinkled yellow type of seeds. Again there are 3. Again there are 3. 1, 2, 3. We have wrinkled yellow seeds. These type of plants will produce wrinkled yellow seeds. And lastly this one. This will be simply, this is only 1. I will write 1 here. It will be green, wrinkled green. It will be wrinkled green. Wrinkled green. Just observe the cross carefully. Until and unless you will not observe it, you will not get it. This 9, 3, 3 and 3, it's actually the phenotypic ratio of dihybrid cross, which means if we are taking 16 plants, we are taking total 16 plants. Out of 16 plants in F2 generation, 9 plants will produce round and yellow seeds, 3 plants will produce round and green seeds, and another 3 plants will produce wrinkled and yellow seeds, and only 1 plant will produce wrinkled green seeds. I simply am saying when we take 16 plants, but number will vary. Whatever the number is, Mendel took that time, or whatever the number we will take or we are taking this time, phenotypic ratio will be same, 9:3:3:1, until and unless we are going through Mendelian genetics. Until and unless we are going through Mendelian genetics. In case of monohybrid cross, what was there? Simply phenotypic ratio was 3:1. 3 tall plants and 1 dwarf plants. But I told that time that number will vary but ratio will be same. Same applies to dihybrid cross. If you are taking 1000 plants, you will see ratio will be in the same form, 9:3:3:1, in which round yellow seeds producing plants form the 9 portions and round green producing seed plants will be forming 3 portions, and wrinkled yellow seed producing plants will be 3 portions, and 1 portion will be wrinkled green seeds producing plants. This is the phenotypic ratio of dihybrid cross. And if you want to go through the genotypic ratio, it will be like this. It will be like this. It will be 1:2:1:2:4 1:2:1:2:1. 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. This will be the genotypic ratio. That means what will be the genetic combination of this dihybrid cross when F2 generation will be formed by self pollination. We will have genotypes in this ratio. 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. I hope you got the dihybrid cross. Please and please go through it again and again so that you will get it what actually Mendel went through at that time. Rest inshallah we will discuss in the next lecture. Till then, stay safe, stay good. Thank you.